Okay, great. So good afternoon from Chicago. However, I do understand that folks may be joining us from any number of places. So I'll say also good morning and good evening if you are in any of those time zones. My name is J.M. Conway, and I am Manager of Innovation Programs with the University of Chicago Grant School. Today, we're so excited to welcome you to our lecture, A Renaissance of Biological Discovery, and updates from Woodhull with Nipam Patel, who is Director of the Marine Biological Laboratory. The Graham School is very proud to be the home of lifelong learning at the University of Chicago. We're actually one of 3 founding divisions at the university because when William Rainey Harper, pictured on the right of the screen, and John D. Rockefeller to his left were imagining the University of Chicago, they wanted to make sure that the big ideas of the university reached all learners at all ages and stages of life. And so they created the first extension school in U.S. higher education. Our school is aimed at bringing liberal arts to lifelong learners. And for 130 years, we remain trailblazers in lifelong learning that aims to rigorously explore the big ideas that challenge and change the world through programs and classes in the arts, sciences, and society. Today, we are joined by Dr. Nipam Patel, who's the Director of Marine Biological Laboratory in Woodhull, Massachusetts. I'm going to turn the stage over to him in just a few moments after I read his bio. Nipam Patel is the Director of the Marine Biological Laboratory and a professor at the University of Chicago. He joined the MBL in 2018 from the University of California, Berkeley, where he was professor and co-chair of the Department of Molecular and Cell Biology. He is a leading scholar in modern evolutionary and developmental biology, with specific focus on the evolution of body patterning and segmentation, regeneration of the germline, and structural coloration. His scientific expertise encompasses the development of novel genetic model organisms for biological study, which can reveal much about human biology, and the application of advanced imaging technologies to probe the fundamental dynamics of living systems. Patel is the 20th scientist to serve as MBL Director since its founding in 1888. And we are so pleased that he is able to join us for today's talk and lecture. I'll share the screen with him. He'll give his remarks, and then we will have time for a Q&A, which we hope you will engage us with. So I'm going to stop sharing my screen now and then turn it over to Nipam. Nipam, thank you so much for joining us.
Thank you. Welcome to everyone, and thanks for this opportunity. So I'm going to, take my time to introduce you all to the MBL. I realize that based on who I see on the screen, there's some people that are very familiar with the MBL or even here at the MBL. But I'm going to, direct my presentation towards those of you that don't know much about the MBL, and sort of explain a little bit about our history, about the research we do. But mostly I'm going to focus on the unique way in which the MBL combines research and education. And education's always been a big mission of the MBL. And I think that we are increasingly eager to have additional opportunities to really educate people at all levels. I mean, we in the summer have huge numbers of Ph.D. students and postdocs here taking courses, but we see great opportunity in expanding that education mission throughout the year and reaching, for example, high school students, or reaching the engaged public who might not have a science, who might not necessarily have a PhD or something like that, but really are keenly interested in science and understanding by being able to do some hands-on work in the lab and to get to explore the kind of unique environment that the MBL provides for that. So with that, I'm gonna go ahead and start my screen sharing and get set up here. Okay. All right. So just a little bit for those of you not familiar with the MBL, where are we? So obviously many of you might be in Chicago, but the MBL is located about 1,000 miles away on the Atlantic coast in lovely Woods Hole, Massachusetts, out on Cape Cod. Here's a view of Cape Cod, and we're out over here on kind of the heel of this boot here. So the MBL has been an institution that is existed for a long time now. So it was founded in 1888. So the original science institution here in the Woods Hole area really came from the Fish Commission, and it grew into basically the fisheries of NOAA. And that started in as far back as probably 1885. And then the MBL itself was incorporated in 1888. And it actually has long ties to the University of Chicago. So in fact, Otis Whitman, who was the first director of the MBL, in fact, became a University of Chicago faculty member. And so there's been these long existing ties actually between the MBL and the University of Chicago. But why have Woods Hole where it is? Why are we located where we are? Is there something special about that? And there is. So this is sort of an ideal place to situate a marine lab because of the interesting environment here. And what I mean by that is that this is a place where warm and cold ocean currents meet, and we are in many ways then for many organisms the northern boundary for many southern species and the southern boundary for many northern species. And because of this environment too, there's very nutrient rich water here, which is why there's a lot of biodiversity. So if you're really interested in studying lots of different marine creatures, this is an ideal place to come. And the next slide just sort of shows you, some of the diversity that we have here, from, interesting, you might be surprised, corals that live here to, of course, this is the feeding ground for whales. Not necessarily that the MBL itself studies a lot of whales, but it's a great place to come see the whales. Anyway, and a whole diversity of organisms, large and small, that are available here as a marine lab. So ever since its founding, though, it's really been a place that combines research and education. So it was founded really by a society of women naturalists at first, and then was sort of taken over by a group of scientists that were resident here. And it always prided itself on being, as I said, an institution that both was a research institution and a education place. Now, there was a time in its history when it considered going just as a pure research institution, but that really was something that, in fact, with the director at the time, Whitman, rallied against. And so I think really a key to success of the MBL has been this combination. So that tie to the University of Chicago became much more formalized in July of 2013, when both institutions actually agreed to sort of unite, and we are now really a part of the University of Chicago, and this was through the vision of Bob Zimmer when he was president of the University of Chicago. So we have a really storied past because many great advances in biology have been made here at the MBL. So in fact, we like to discuss how 60 Nobel Prize winners are affiliated with the MBL. Now, obviously, we have to be careful how we use the word affiliated. Some of these are just people who've come here to teach in the summertime, for example. But in many times, there have been very key and big discoveries in biology that really were made at the MBL or really blossomed at the MBL. So these include very basic discoveries like how your body, how nerves conduct impulses along their length, or the fact that you can use horseshoe crab blood to detect bacterial contamination, a test still used to this day to validate medical instruments and vaccines. And then it was here at the MBL, the discoveries were made about the proteins that control cell division and the lack of that control in cancer, and then how molecules are transported within cells, just to name a few of the remarkable breakthroughs that have happened over the course of the MBL. So as I said, the education started, classes started with the founding of the MBL. Here's one of the older classes at the MBL, embryology in 1893. You'll notice that there's also quite a number of women in the course. That's one thing that MBL could take some pride in, is that it included quite a number of women in the early classes. But it was really, like I said, the thing that was unique about it was a place where active researchers were really taking the lead in training the students, and in a training that was very hands-on. So it's not based on lectures, it's based on actually doing experiments, but not just rote experiments. It's based on challenging students to make new discoveries and to push forward. So here's maybe a slightly more modern picture of that same course, embryology from 1979. But again, it shows you the kind of people that took the course. So for example, Tim Hunt, who went on to win the Nobel Prize in 2001, was in that course. Joan Ruderman, who went on to be the first female MBL director. And this guy here, Jerry Rubin, actually was for many years helped establish Genelia and was director at Genelia, the Howard Hughes Medical Institute's research arm. So to this day, we still in the summertime run a huge number of courses, about 21 courses each summer in a wide variety of areas, and that's sort of illustrated here. And so some strengths include the courses we run in development and reproduction and neuroscience, all areas of cell physiology and microbiology, and then a long-standing excellence in microscopy. So the courses really have changed over time, but they've kept up with these different fields. And so in a typical summer nowadays, we will have about 500 students. Most of those students are graduate students and postdocs who come here in the summertime, and then about 500 faculty who come and teach. And then in addition, we have about an additional 300 scientists that come to do summer research. And these courses, we think, and I think there will be plenty of people that would agree that have taken these courses, are really some of the top courses in these areas of biology. And we ask students for feedback, and interestingly enough, on our scale, the highest scale for rating things that happen here is transformative. And it's amazing the number of students that check that off. And again, it's because it's not just following a cookbook, it's actually doing original research here during the summer. And that's really what's led to many of the amazing breakthroughs. So, for example, there's something called the Breakthrough Prize, which was just awarded last year. And one of the co-awardees was Cliff Brangwine. And he and his other awardee, Tony Hyman, really made their breakthrough discovery of how certain sets of molecules are organized within cells here during the physiology course. So it's a great example of that tie between the research and education that goes on. Another reason that we do extremely well with our courses is the amazing cutting edge equipment we have for basic biology research. So some of it we build and have here, and other pieces are lent to us by vendors. So for example, the microscope companies every year will bring their latest and greatest here to the MBL because they know this is an excellent place to test out their equipment, and they know that amazing discoveries will be made with that equipment. We have a building dedicated to education. So this is called the lower building. And in it are these very large classrooms that really are designed for teaching. And I've taught in them now almost 25 years. And they really are the perfect place to sort of run courses. Aspect that is responsible for our successes, again, of course, that we are a marine lab, we're on the ocean. Now, we don't exclusively do what you would call marine biology. Many scientists here do basic biology on, cell cultures and all sorts of things like that. But we do very much take advantage of the fact that we are a marine lab and have access to all of these different organisms that live here. So we have a boat called the Gemma that goes out all the time and helps to collect animals. And then we have a very large building called the Marine Resources Center that has flowing seawater and systems for maintaining marine organisms. So we can collect all manner of organisms that researchers may want for either research or education, house them within the MRC and have them available. And again, this is just examples of some of the animals that we go and collect. Now, we also have a resident research program, which is a very vital part of the MBL. So the researchers are here year-round doing research, and they can be divided up into a couple of different areas. So we have an ecosystem center that's very much devoted to the study of ecology and climate change, has a particular emphasis on looking, for example, in our shore environments here, at what we can do to preserve those environments against climate change. We have a lot of biological discovery in marine organisms. I'll talk a little bit more about that, for example, many marine organisms are capable of incredible feats of regeneration. And so we have a number of resident scientists that are here year-round working on those kinds of problems. And that's primarily in a center called the Bell Center. And then we also have a number of resident scientists that focus on microbes and their interaction with in organisms and in the ocean, the planet, and for human health. And a lot of that is research that's located in our Bay Paul Center. And then, as I mentioned before, we also have had a long-standing strength in microscopy and imaging. And so that really is something that runs through all of the different areas of research that we do. So since I've been here, there have been two sort of strategic initiatives that I've been involved in trying to really further the strengths of the MBL. So both of them take advantage of long existing strengths. One is the development of a variety of aquatic, mostly marine organisms for biomedical and basic research. So you may all be familiar with the fact that often scientists really focus on a relatively small number of organisms in which they do biological discovery. And these include things like the fruit fly or the nematode worm, mice, and zebrafish. Now, these organisms are so-called model organisms for a good reason. So they usually have a relatively rapid generation time. They're inexpensive to raise. And because a lot of people have worked on them now for a long time, they have many tools or approaches or techniques that have been worked out to do all manner of experiments. But there's a problem. So these animals represent only a very tiny slice of the diversity of life on Earth. And in fact, there's lots of good reasons to really expand this repertoire. In fact, a long time ago, before these models were dominant, people did work on a vast variety of different organisms, including lots of marine organisms. But then over the years, people in there, have taken advantage of these model organisms to really focus, and for good effect. So a lot of big breakthroughs have been made in them. But there are now many of the techniques that have been developed are really applicable to any organism. And so there really is now a renaissance in really opening back up and studying a variety of organisms. And so why expand this? Why especially to marine organisms? Well, one is to understand how evolution has generated diversity and the mechanisms of evolution. That's something that is very near and dear to my heart and a couple of the researchers that are here as resident researchers or visit during the summer. But there are other reasons. So for example, so we really want to understand that evolutionary history, how we're related to other organisms, what things are conserved in evolution, what things were responsible for evolutionary diversity. But there's also a lot of interesting biology that's not well represented by our current major models. And a really good example is regeneration. So many of the organisms that most scientists work on, in fact, are not that good at regenerating. But there are many, many aquatic organisms, especially marine ones, that are great at regeneration. So one of the ones that we work on here, an aquatic, a freshwater organism, is the axolotl. So this may be one of the masters of regeneration. So you can cut the limbs off of axolotls and they will regrow a perfectly good limb in a matter of weeks and months. And so, of course, we would love to know how the axolotl does that and use that information to, of course, help in situations where humans need to regenerate organs or limbs or whatnot. And so we and others are using these kind of marine models to do that. So axolotl is one. I also work on a crustacean called parhyala. It's a little beach hopper amphipod. But like many crustaceans, you can cut the leg off. And in the case of parhyala, within about 2 to 3 weeks, they will have grown a perfectly good leg that looks exactly like the original and functions every bit as well. So again, studying these organisms can have both profound advances in basic biology as well as translation into thinking about how to apply that to human health. Likewise, studying things like corals, we know that this is a dire problem now for the world, is the deterioration of coral reefs. Many will be surprised to know that there is actually a coral that grows here in this area. And it's one that you probably all heard about coral bleaching, where coral expel their symbionts, but the coral that lives here in the Woods Hole area can actually, when it needs to, take up symbionts, and when it doesn't need to, it can expel them, but can naturally take them up again. So this is an active area of research, is trying to understand, can we understand how this coral works in order to apply it to saving other coral. So as I said, expanding this area of the different organisms that is used is actually one of the innovations. Another way that we've expanded that is really to be a world leader in studying cephalopods, so things like squid and octopus and stuff like that. Why study them? Well, one of the reasons is we pride ourselves as being human invertebrates of having evolved very, very sophisticated brains. But in fact, cephalopods, things like octopus, are one of the other lineages of animals that's really been able to evolve a very sophisticated brain and then display very sophisticated behaviors. So a number of our scientists are really interested in understanding how it is that you fashion the brain of a cephalopod and what is similar to our brain and what is different. Cephalopods are also fascinating because they carry out a phenomenon known as RNA editing at a much higher level than other organisms. So many of you may be familiar with the idea that DNA encodes our genetic material, and that is read as a molecule called RNA, and usually read very faithfully and then read into proteins. But in fact, cephalopods are able to do something called RNA editing, where they can change these base pairs in the RNA, and that leads to different proteins. And there's some tantalizing evidence that this may be responsible for some of their remarkable adaptations. And so we have scientists studying that. Why is the MBL a great place to expand the repertoire of animals? Well, one is that we have a history of success in developing these kinds of different creatures for basic biomedical research. And so we build upon that strength. And the other is that most of the diversity of life in the world really lies in the ocean. So if we really want to examine that diversity, being a marine lab puts us in an ideal position to do that. So what are some of the challenges? Well, one is husbandry, being able to raise all of these different creatures and then so that scientists can take advantage of that. And that's, again, where our Marine Resource Center is one of our strengths, because we have this capacity to culture native species, but we've also recently been increasing our capacity to culture tropical or non-native species, because many of these have much faster generation time. So for example, an octopus will take years to go through development. But here at the MBL, we've now cultured quite a number of different cephalopods, including tropical ones that can undergo their whole life cycle in a much, much more speeded up way. And so we can apply many of the tools of genetics and genome editing to those animals. So in fact, that's the other thing we're doing, is developing the necessary tools to make experiments possible in these different creatures. So for example, inserting genes into these animals to track what cells do. So we can insert a fluorescent green protein into axolotls that lets us see mechanistically how different parts of the limb are regrowing during their process of regeneration. We've also been working out techniques to manipulate organisms at the level of DNA editing. So many of you have heard of this phenomena of CRISPR, where we can go in and edit the DNA. And this just again shows in the animal I work on, Parhyla, these little beach hoppers, we're studying how they make different kinds of legs, and we can CRISPR individual genes and actually change the kind of legs that they develop. And so this allows us not only to do basic research and understanding how crustaceans have diversified their legs, but these same genes, in fact, are involved in setting up our spinal cord. And so the information that we gather is directly relevant to understanding human health as well. And finally, we've been making inroads in imaging in these different research organisms. So we've developed microscopes that do things like allow us to image coral while it's still alive. So normally coral is very sensitive to light and will pull back, and it's hard to see what coral is doing at any given moment using sophisticated microscopes. But we've been working on techniques that allow us to do things like that. So in addition, then, as I mentioned, we have a number of scientists that are really interested in the microbiome. So you've probably seen a lot of publicity over the years, recent years now, about the realization that our microbiome, those microbes that live, for example, in our gut, are absolutely essential to our health. And that they were, often thought of as, well, just being disease causing. But in fact, we understand that they're very important, not just for human health, but the organism of the health of all organisms and the health of ecosystems, because they help, they're essential to transferring nutrients and to signaling in all sorts of environments. And so one of the environments that many of our ecosystems scientists study are coastal environments, where they're looking at how all of these microbes and plants and everything interact to set up those unique situations, or so unique, like, for example, seaweed beds that protect our coastlines that we desperately need to preserve. So what we've done, as I've said, is we've incorporated all of this not only into our research, but into the teaching that we do in the education. So for example, all of the courses we teach take advantage of this diversity of organisms. Likewise, with like our imaging technology, we actually, when we bring students in to teach, and that is all the way from, these Ph.D. students to the public that we bring in, and high school students that we bring in, we actually put them onto these sophisticated pieces of equipment to do things. And so in courses that we do, and things that you might have an opportunity to do if you're take advantage of this ongoing education that we offer, are things like learning how to do CRISPR. So this technique of editing genomes, which holds a lot of potential, but also some ethical dilemmas for treatment of human diseases. So here at the MBL, we have, for example, high school students that come in and they do these experiments in zebrafish. They actually make mutants in the course of 3 days and are able to see the results of their work. They get to see how these embryos develop. Here's a time-lapse movie of zebrafish embryos developing, and you can see the embryo going from just a unpatterned set of cells into, what clearly is now an embryo with an eye and the muscles growing down its back and everything. Again, as I mentioned, we teach, students how to use very sophisticated imaging equipment, and then we'll teach, any of you, if you're interested, how to put that onto your computer, make 3D reconstructions, and really be involved in advanced imaging, both of these sort of fixed specimens to look at how muscle and nervous system are made, in this case in zebrafish and in flies and in squid, and then even doing things like live imaging and understanding how blood vessels are set up in something like a fish. Students who take courses there also have an opportunity to engage in the ecosystems research that we do, so to travel out to the marshes that are really right here on our doorstep, and to get a chance to see and you do with your own hands some of the experiments that our ecosystem scientists do to look at nutrient cycling, to look at the interaction of different organisms in these environments. And then within the microbiome center, to be able to do things like isolate these microbes and then do DNA sequencing to understand what these different organisms are and how they interact with one another. So I hope with that, I've given you a very brief, but hopefully, useful overview of both the research and education that we do. And as I said, this has been a place that's devoted to an equal mix of research and education. And I think it's a really unique environment to come and bring, people who are engaged in lifelong learning, who want to really work with scientists to do not just, some cookbook experiments, but really get a chance to see and try, in fact, to solve fundamental problems in biology. So with that, I'm happy to take any questions.
Wonderful. Thank you, Nipam. I do have some questions that have already begun to gather here in the chat. And so I may just go ahead and read maybe a couple at a time and allow you the space to answer those questions. So Timothy asks, is there something open for the public there at the MBL? I imagine exhibits, museums, tours, and trips on the boat. And then we have a question from Janet that asks, can you talk a little bit more about the students and also the teachers?
Okay,
How are many universities? Oh, okay. I just want to read the rest of the question here.
Okay.
How are many universities from the many countries from how many universities, from how many countries do they represent? Are there scientists outside of academia? And the questions are also in the chat, Nipam, if you want to.
Yeah, and some of them. Yeah.
Okay.
Okay. So let's start with about the tours and things like that. Yes, we do. In the summertime, we have tours. You can sign up for the tours, and then you're able to go through see some of the facilities we have here, like the Marine Resource Center and some of our public exhibits. And I think, it's a great way to learn a little bit about the MBL and to see some of what we do. We are increasingly, looking at having courses that the public can take. And so one is obviously what you'll hear about, the possibility through the Graham School for Lifelong Learners. And then we've really in the last couple of years launched a whole set of high school courses. So high schools from around the United States come and participate in these courses. So we do it for local public schools, we do it for private schools that come from far away, but it gives students a chance to really do that, and teachers as well. And then what about the summer courses? So I think we have as many as 130 different institutions represented from around the world that have come and sent students here. And so it's really remarkable. In fact, it's fantastic to be here in the summer and hear all the different languages that are spoken and the incredible diversity of people that come from all over the world, really, to take our courses. So it's fantastic. And are there scientists from outside academia? So we do have some courses like the imaging courses that we run that often scientists who work at companies will come to. And increasingly, of course, there's interest for some of the biotech companies that are in the United States, especially, to come and have some training here. And certainly many of the students that get trained here, some of them don't go on necessarily in academia, but instead go to biotech or they become writers. And in fact, we run a course just for journalism students, for example, so that they can learn about the science that we do. So we really try as much as possible to have a broad education mission, not just to academia, but to all areas of society.
Thank you, Nipam. Another question here, how is the MBL and its programs funded?
So it's a mixture of federal funding. So we have funding for our research from the National Science Foundation, the National Institute of Health, and then things like the Department of Energy also fund some of the research that we do. Our education missions are funded by, again, from the federal government side, by the National Science Foundation and NIH, but also generous funding from private foundations. So the Howard Hughes Medical Institute and the Burroughs Wellcome Fund also provide significant funding, and the public. So there are endowed lectures that have been funded by the public and things like that that make our courses possible. And foundations also are very important to often providing the seed work for research before it, has enough results that NSF or NIH will decide to fund it. That's sort of a critical thing, especially for young scientists. So we get significant funding, again, from a lot of philanthropic foundations and from donors as well.
Thank you. Here's another question from Janet. Thinking beyond scientists, what is the takeaway core message of the MBL? Many people know about Rachel Carson's affiliation and high regard of the MBL, but her books were published decades ago. While we're talking about her, is there research on green chemistry?
We really focus on biology, but there is, this amazing interest though in understanding, for example, how microbes carry out complex chemistry and, the reduction of waste and things like that. So people within both the microbiome side of things and the ecosystem center are very interested in how we preserve the environment, how we can come to biological solutions to some of those things. But of course, biology is based on chemistry. So understanding the chemical processes that guide that, for example, getting rid of nitrogen waste and things like that are all things that our scientists, are interested in and do research on.
Joe Wesley asks, if one is visiting Woods Hole, is there a good way as a tourist / civilian to learn about current research, e.g. a museum, the current findings and research presentation space?
Yeah, I think that, there are a couple of ways. So one is, for example, we have lectures every Friday evening. So we have notable scientists that come here and then they give a large public lecture, and then you have a chance to interact with them and the rest of our scientists in that. And then in the non-summer months, we actually also have a Falmouth Forum. And increasingly, we've been engaging a lot of that online as well. So even when you're not here at the MBL in person, you can join us online for those lectures. So I think that's a great way to learn more about the research that goes on here.
Great. Another question, does the MBL research include principles of origin of life and astrobiology? Does the MBL collaborate with NASA? How ocean research can advance our understanding of the origin of life?
Yeah, I think, well, we certainly have summer visitors that are interested in the origin of life. It's not something I think that, is a main focus of our resident faculty, but of course, understanding how life has evolved in the ocean, which something like, actually NASA is very interested in, because those principles are important in understanding how life may have evolved on, on other planets. So while we don't necessarily look, for example, at how the first proteins may have been formed or something like that, we are interested in how life is diversified. And so that is of fundamental importance in understanding the possibility of life on other planets.
Thank you. Jerry asks, you showed a slide that displayed the interaction of the shelf break jet and Gulf Stream that increased biodiversity. Is it possible to measure things like that in order to explain, the name of this animal is hard to pronounce.
Axolotl, yeah.
Axolotl regeneration. For example, measuring the flow of blood or oxygen, general culturing conditions, which must act in concert with genetic mechanisms.
Yeah, it's a great question. So at first you might think, well, wait, what do these have to do with each other? How does, the flow of water at large scale have to do with something like regeneration? But it does really help to illustrate an important principle, which is that, in fact, physical forces like this have often been ignored in especially in more recent biology, which has taken a very genetic focus. But in fact, we know that the physical interaction of cells and the tension that the cells have and the frictional forces as they try to move by one another actually is very instructive to the cell. And gene expression itself is often guided by feedback from those forces. So in fact, there are an increasing number of scientists who really have a training from physics who are getting very interested in biology because they can apply those lessons of physics modeling what actually goes on in biological systems. And you're absolutely right that in fact, there's a lot of interest in things like, well, you don't actually form a blood vessel, right, if there's not actually flow in the vessel. So those physical forces are actually vitally important to the biological outcome.
Thank you. Clinton and Karen ask, what are you observing in terms of climate change, sea level rise, and so?
Yeah, so there's both the threat to the MBL itself as the sea level rises, because we're right on the water, so we see that. But there's a lot of interest in understanding what's happening to the marshes as the sea level rises. What's going to happen to those marshes? Are they going to respond fast enough? Are they going to move back into higher ground, or will the sea level rise too quickly for that? And what can we do about that? So those are things that we are in fact observing those impacts. And I should point out that MBL scientists work not only on local marshes, but they work in a variety of systems. So there's also a long-term ecological monitoring site at Plum Island, which is north of Boston, as long as some of our scientists who have long-running projects in Alaska and looking at the ways that climate change is affecting those environments and what we can do about that. So it is something that we both see the impact of on our local community and are concerned about and are thinking about. And we, for example, we're part of a consortium, the Woods Hole Resiliency Group, which is really looking at how we protect not just our institutions, but the entire village of Woods Hole against climate change.
Thanks, Nipam. That actually relates to perhaps another question that we got from Janet. How does the MBL interface with WHOI and NOAA?
Yeah, so there's 6 science institutions that are here in the Woods Hole area, and the question mentions 2 of the other larger ones, the WHOI, the Woods Hole Oceanographic Institution, and NOAA. So we do interact quite fruitfully with both of them. So WHOI has lots of, for example, large ocean-going vessels, and so some of our scientists will take advantage of the resources that they can get by going on those trips. But then also WHOI, for example, will use many of our facilities. So they use our electron microscopes and light microscopes for their own studies when they are doing those kinds of projects. And NOAA is right across the street, and we and NOAA sometimes collaborate on things. So for example, we have a small project looking at migration of some of the striped bass that come to Eel Pond each year, which is an interesting behavior for these striped bass. And NOAA, experts from NOAA have been helping us think through some of our projects on how we would understand the biology of those striped bass.
Okay. I'm just looking at our time here, and there are a few more questions that have gathered here in the chat. I do want to reserve a little time to share with you all here who have joined us a little bit about our program, an upcoming program in partnership with the MBL and the Graham School. So perhaps I will ask one more question, and then we can move on to that announcement. Let's see. I think this might be an interesting one to end on. Janet asks, what will the MBL look like in 10 years?
Well, hopefully some parts of it look the same. We have some beautiful historic buildings, so we don't want those to change. Hopefully we have mitigated against some of the impacts of sea level rise. But we do hope to make some renovations to the insides of our building and to set up a new imaging core, to expand and improve our housing and dorm structures and things like that. So I think, we have a beautiful historic look. We don't want that to change, but there are a number of buildings which I think that you're going to see even more and more modern labs inside of. So we're looking for a very productive period coming up. I think we're going to see expansion of the MRC to be able to hold more and a greater diversity of marine creatures, and we're working on automated systems that raise some of those. So I think we might look pretty similar from the outside, but from the inside, we're going to keep growing.
Thank you so much, Nipam. And I would invite others here to share your gratitude for Dr. Patel's lecture and Q&A segment in the chat if you'd like to. add some gratitude there, that'd be appreciated. And of course, we always thank you, Dr. Patel, for joining us. I'm also now going to share my screen again to share about a really exciting opportunity along the lines of many of the questions that some of you have posted in the chat around learning at the MBL and a special opportunity to come and be with us in Woods Hole. So are you all seeing my screen? Okay, great. So as we close out today's lecture, I'd like to take a moment to tell you a little bit about a special learning opportunity to study in Woods Hole at the Marine Biological Laboratory. Through a partnership between the Graham School and the Marine Biological Laboratory, we are offering a scientific discovery program in which you'll spend 8 days living and learning alongside faculty from the Marine Biological Laboratory's 3 research centers: the Josephine Bay Paul Center for Comparative Molecular Biology and Evolution, the Eugene Bell Biology and Tissue Engineering Center, and the Ecosystem Center. This is a hands-on program where we are inviting students from all ages and stages to learn about and experiment with cutting-edge technologies in gene editing, ecology, and more. And actually, these pictures on the right of the screen are pictures that I actually took myself while in Woods Hole last year. They show some pictures of participants engaging in micro-injections as practice for an upcoming experiment with zebrafish mutations. So Nipam mentioned that earlier. The students in the program last year actually got to do some of that experimentation while in Woods Hole. And I actually see some of the participants from last year's program on the call. So hello to you all, and it's good to see your faces and names in the place. We also got to spend a little time on the Gemma. Nipam mentioned the Gemma. While we were there, we got to go out and collect some marine organisms to see close up. We took a visit to the marsh and had other activities throughout our time together. It was a really enjoyable time there in Woods Hole. And so as we close out today, we'd love to welcome you to take a look at this program on our site, the Graham School at uchicago.edu. The tuition and fees for this program come in at about just above $4,000. That includes a virtual orientation, program faculty, as well as your particular learning cohort for the year. It includes those 8 days of learning and experimentation in Woods Hole. Also those associated activities and events. We had various folks asked about tours and lectures. There were tours and lectures. We took a tour in which I got to actually hold a Nobel Prize, which was really exciting, as well as a rare books tour in which I saw a book that was actually signed by, I'm going to look for the name of the person who signed this book. I have my notes here. Charles Darwin actually signed that book that I held. So that was actually really, really cool. So it includes activities like that, as well as other activities on the Cape. And then finally, it includes the on-campus room and board. So we want to thank you all so much for joining us today. I will take a moment before we end the call today to add the link to more information about this program. And we will also message you with more information about how to get signed up and to answer your questions about the program. So once again, thank you all for joining us today. And we hope to see you perhaps as a student with us in Woods Hole in the coming months. Thanks for joining us.